Okay, moving on to the next section, section 3. So now we're going into the clinical manifestations of the infection of the disease. Right, so the actual manifestations can vary depending on what the pathogen is. Some, like we saw before, don't have to show any clinical symptoms. You can have bacteremia and not show any clinical symptoms. At the same time, some disorders, the diseases come out really quick, like the physical symptoms. Something like anthrax. Within the first day or so, you're going to start having this pulmonary edema. It's going to feel like you're drowning in the lungs. Something like cholera, within the first day or two, you're going to have the same problem, except it's diarrhea. So the water's being excreted into the GI tract and pushing out. Other ones are going to cause a fever. So most infectious diseases, almost all that I could think of, trigger some type of fever. It's your body's response to get rid of it. And we're going to talk in just like a slide or two about the fever. It may be the chemicals that are in the bacteria that are causing the fever, but it may actually be your white blood cells that are releasing chemicals to cause the fever. And when you want to think of fever, you want to think of a certain part of the brain, and I'm hoping you're already thinking what part of the brain is going to control your temperature, or your freezing, your feeding, your fighting, your fornicating impulses. And that's the hypothalamus. Right. So, give me fever. Fever is also known as pyrexia, and you take an antipyretic to reduce the fever. And I found this little slide, or this picture over here, to put on the slide that's of the process. So here you can see what's happening, and I said before there can be two causes. It can be the actual bacteria itself, so like an endotoxin from those gram-negative bacteria being released into the system. When a white blood cell destroys that gram-negative bacteria and releases this endotoxin, that endotoxin can circulate and become a pyrogen itself. Right? So it's considered a bacterial endotoxin. This is where it gets a little bit tricky, because when you refer to an endogenous pyrogen, you're actually talking about something released by the body, your human body. When you're talking about an exogenous pyrogen, it's released from the bacteria or the invader. So an endotoxin can be an exogenous pyrogen. So it stimulates the fever, but it's from outside of the body. So in this situation, this bacteria here, when it's destroyed, it releases part of its cell coating. It goes up to the hypothalamus, and the hypothalamus is going to start releasing prostaglandins. The prostaglandins are going to reset that temperature set point. So if your normal body temperature is 98 degrees, it bumps up a little bit higher. Basically, it's your body's way of saying, hey, there's a bacteria in here. The bacteria doesn't like to replicate outside of its little tiny comfort zone. It's not like you, where we have a lot of fat. We can adjust our temperature. If we get a little bit cold, we can burn some of that fat for energy. If we get a little bit cold, we've got that padding that helps insulate us. A bacteria is one single cell, has that thin wall. If the temperature gets too hot or too cold, it doesn't survive very well. And at least, at the minimum, what it's going to do if you take it out of its comfort zone is it's going to slow down its replication. And that's kind of the key. When you heat up the temperature of the body, it slows down the rate this is going to replicate. So it gives your body a chance to send the white blood cells in the area and attack these. Right? So here you have tissue that's being infected by the bacteria. The white blood cell comes in. So they're going to come in there. They're going to start eating the bacteria. They're going to release cytokines. Remember, those cytokines are the chemicals that are telling other white blood cells come into the area. Well, some of those cytokines are actually going to go over, like interleukin, ILs. Remember those? And they're going to stimulate the production of prostaglandins. What's interesting is that prostaglandins can actually be stimulated at the vagus nerve, and it sends a signal up to the hypothalamus. So prostaglandins send a signal up to the hypothalamus, okay? Or the prostaglandin can penetrate through the blood-brain barrier, and they're going to start triggering part of the hypothalamus. And this POA-AH means preoptic area, POA, and then anterior hypothalamus. They call it that because it's really close to the optic chiasm, and it's a long story you don't need to know. But basically, that just talks about part of the hypothalamus that's being stimulated. The hypothalamus raises its set point from 98 to 99. And as long as there's still bacteria and there's still white blood cells attacking the bacteria, you have these pyrogens that are circulating through your system. They keep raising it from 99 to 100. Well, if that didn't kill off the bacteria, we need to go up to 101. If that didn't kill off the bacteria, go up to 102. And it's raising and raising to help you to slow down the bacteria and possibly kill the bacteria. So your body, fever is a good thing as long as it's controlled. When it becomes beyond control and it goes over 102, then you have to start worrying. So when it goes to 104, you should get getting your way to a hospital, so they can help regulate your temperature. Because if you hit 108 to 110, you're dead. You can't live beyond that. When your blood heats up so high, your proteins start coagulating, your nerve cells start shutting down, the cell membranes start destabilizing. You're falling apart, basically. You're circling the drain. So you need to keep your temperature low enough that you stay alive, but high enough that you kill off or stop these bacteria from spreading. And that's what a fever is all about. When you look at the symptoms, what's interesting is when you look at the pathway, and we're going to talk about this a little bit more next week when we talk about the nervous system, or the week after when we talk about nervous system. It's one of the 2 weeks of central nervous system we talk about it. But you have to think about the symptoms. When you're generating the fever, you feel cold, you have the shakes because your new set point's at like 101. And even though your body temperature is 100, it thinks, I got to get up to that 101. So you start shivering to generate energy. You start shaking all over the place. You start covering up with a blanket to try and crank your heat up. And then once you got rid of this bacteria, now the bacteria is gone because you heated it and you killed it with white blood cells. Now suddenly your set point says, well, we're not getting signals. The bacteria must be gone, so let's go ahead and turn that back down. And it turns it back down to 98 degrees, but by now your temperature is at 101. So now what's your body think? When your fever breaks, suddenly you go from that freezing, shaking, pale skin experience to suddenly you're really hot. You're sweating, you're ripping the covers off because it's too hot, and you're trying to cool down. You're trying to get your body from that 101 back to its normal, healthy 98 degrees. And that's fever. And we'll talk about, again, when we talk about the brain, we'll talk about the fever. Right? So other ways that we strike back. How do we get back at these bacteria? Right? So here are some countermeasures that we do, or actually some preventative measures. So a few different ways is, well, one, give vaccines. And with vaccines, what we're doing is we're trying to create memory that we talked about last week. You give yourself a little piece of the antigen, or, well, I should say the doctor gives you a little piece of the antigen, and it stimulates your immune system to start turning up, to start making these memory cells. It's going to make B cells, and hopefully you remember what the B cells are going to make. So once they're activated, they turn into a special cell called a plasma cell. And once the plasma cell is activated, it makes tons of what little structures? Antibodies. When you activate the plasma cell, you also create a second B cell that goes and hides in the closet. Remember, that's how I talked about it last week. It hides in the closet so that later when you're re-exposed to this thing, it can come out of the closet, it remembers the war the first time, and now it gets a fast response. It's a memory cell, so a memory B cell. So vaccines are there to help create memory. Right, here are a couple of different types. So you can have something called a live attenuated organism. So it contains the bacteria or the virus, but it's a weak version of the virus. So if this bacteria was a bad bacteria that can make you really sick, what they've done is they've made it so that they can't hurt you anymore. They've taken away whatever genetic property it had that made a toxin or made some kind of a poison or attacked your cells, so that it's still alive and they inject it into you. So some good examples are the measles, mumps, and rubella. It's a live attenuated vaccine, and what happens is they weakened it, but it gets in your system says, hey, look at this thing, we know what this is. Now, it starts mounting this immune response towards it. Sometimes when you get an injection like that, the day after you feel really tired, you feel worn out, you feel like you're getting the actual cold, or you're feeling like you're getting the actual disorder. But what it actually is that you're just fighting it. You're not sick. You don't have the flu. You don't have the measles, mumps, or rubella. You're just fighting, basically, an antigen in your body. Right. Another way you can do it is if whatever it is really, really bad, like really dangerous, like the influenza that in 1918 wiped out 30,000 Americans, and I think across the world, like 150,000 people. It was a bad thing. So something like that, they want to kill that. They kill it, they chop it up, they put it in little tiny fragments, and then they give it to you. They want to make sure it's safe. Same thing with polio. So with polio, we take it, it hurt people severely. It left them crippled or dead. And we kill this thing, chop it up into little pieces. I always imagine this little tiny blender. You throw stuff in there and you hit frappe, and it chops into a million little pieces, right? And then you give it to people. Another way to do the killed organism is they'll actually take the organism and they'll surround it with formaldehyde, which kills it. And you work in an anatomy lab where you have formalin, that's a preservative. They give it formaldehyde and it kills it. That's kind of an issue because then suddenly you're injecting formaldehyde into people when you inject this vaccine, so it causes some problems. The next one is toxoid vaccines. And toxoid vaccines, if you're trying to get rid of a toxin, so if, for instance, tetanus. So when the tetanus bacteria gets into you, it throws this little tetanus toxin out. And this tetanus toxin circulates around. You make antibodies against the tetanus toxin. It's the toxin that's dangerous. So with toxoids, they're basically giving you a little sample of this tetanus toxin that doesn't hurt you. It's been made where it's not toxic anymore. Did I skip over any? Oh, recombinant viral DNA. This is when they take bacteria or yeast and they make like large quantities of a single protein from the bacteria. So we know one distinguishing feature of the bacteria or one distinguishing feature of the virus, and we make all of these antibody clones. I shouldn't say antibody clones. We make all these clones of that little piece of protein. And then we give it to you, your body detects that piece of protein, and then you make the antibodies. So that if you're ever exposed to this in the future, you have the antibodies already made. Right? And that's recombinant viral. I think I got them all. So we should be good to go. Sorry, last question. All vaccines are trying to induce what to the immune system to produce antibodies. So every one of these actually introduce a little tiny structure we talked about last week. And it was antigens. They introduce antigens to the body so that your body can mount this immune response against it. There's a little guy, so you're going to grab your pencil and paper. And what you're going to do is you're going to look up these different disorders, or actually these different bugs, we'll call them. And you're going to write down what kind of vaccine we give them. So do we give a live attenuated? Do we give a killed vaccine? Do we give a toxoid against it? And you're going to look up these. So some of these you may be familiar with, like herpes zoster, which causes shingles, or you can look up varicella, which is chickenpox. You look up the measles, the mumps. Right? When we get the flu shot, what is it you're giving these people? I already talked about it before. You're giving 3 different, completely different flu strands. Or what about with pertussis? Or whooping cough? How about with rabies? And I mentioned earlier, if you remember what it was. What do we give to them? And then hepatitis A and hepatitis B, HPV, you can think Gardasil. So what's in that Gardasil? Is it live attenuated? Is it a killed is it a killed virus? And then tetanus. What are we trying to do? So you want to jot those down, take a little time, and you can come back here. Looks like it's about 13 minutes in, and you can write those down again. But you're going to go back, you're going to write down what it is we use for the vaccine. So which of these different options we use when we vaccinate against them. All right, next, antimicrobials. So now we're going to fight back, not the vaccine to prevent it, but to kill it. So what we have to look at with antimicrobials is we're looking at what makes that organism different than our cells. When you look at eukaryotic cells down here versus something like a prokaryote, you can see one of the big things, we have a nucleus, they don't. So their DNA is not as protected as ours is. When we look at how we make energy, of course we have mitochondria, which is the powerhouse, but remember these prokaryotes are about the same size as mitochondria. So they work a little bit different. They don't have mitochondria. What they are very similar to mitochondria, where they use the cell membrane and enzymes in the cell membrane to make energy. So we can look at these things that are different, target them, and try and kill the bacteria using those. Or the virus, if it were antiviral. Or the yeast in this antimicrobial situation. So there are a couple different things that could happen. We can try and stop the bacteria from being able to build a cell wall. If we stop it from building this wall, it can't replicate. Okay? It can't repair the wall. Another thing is maybe survival proteins. Maybe there's some proteins inside here that are being made that keep this thing alive. If you turn off that protein, basically it starves to death. Maybe it's replication material. Maybe there's a special enzyme that helps it replicate. So if we shut that enzyme off, it can't replicate. It gives us a chance to bring our white blood cells along and chop that sucker up. Maybe we inhibit its metabolism. Since we know how its metabolism works, maybe we can target that enzyme on the cell membrane and turn it off so it can't make energy. If it can't make energy, it's going to die. So when you look at some of these different methods, the antibiotics or antimicrobials, when you get into pharmacology, you're going to see that they work on those different methods. And as just an example, I'm going to have you do another quick one. This is pretty easy because this answer is all over the internet. But what I'm going to have you do is Google this, and you're going to find out how Fleming discovered penicillin and how it works. So how he discovered it is pretty famous. You probably have already even heard about what happened. It was we'll call it kind of serendipity. It was a fortunate accident that he discovered penicillin. But don't write me a book on it. And then write, how does penicillin attack a bacterial cell? How does it work? And that's it. So I'll give you a chance to do this assignment, this Google assignment, and going back and jotting down these. So types of vaccines that we use for each of those pathogens.